Well, thank you very much to the hardworking people who have organized this really interesting conference, and my apologies to the mini symposium speaker who got bumped. Thank you very much too. I've been working on dynamics in the nervous system for several decades now, getting more and more into issues of details involving the dynamics and function in the nervous system. But for this talk, I decided to back off a little and talk about a very general mathematical issue, which I think is central to understanding some very important neuroscience issues. So, networks of neurons usually have dynamics that have some oscillatory power. But the networks and the cells that are part of those networks are not themselves what we normally think of as oscillators. And this is very important because any given part of the nervous system generally is getting inputs from other parts of the nervous system. The inputs have some spectral properties. The target has dynamics with spectral properties. And understanding how the target deals with its input is a huge, huge expansion of the kind of theory we have of forced oscillators, in which the target is something periodic and the input is something periodic. So it's hugely more complex. And this complexity, I claim, isn't a side issue in understanding neuroscience. It's in fact at the heart of how the nervous system manages to respond flexibly to its inputs. So that's what I'm going to be talking about, and it's all in the context of what role does oscillatory activity play in brain coordination. See if, this does work, good. So I'm going to be talking about brain rhythms, which are objects that are not well defined like oscillators are. They're squishy. To understand what they are, we start with electrical activity that's produced by the brain, 27, or possibly magnetic activity. And one can see this in single cells. You can see it in local groups of cells, and what they produce is called the local field potential. You can measure it non-invasively with EEGs and MEGs. And once you have this, those signals tend to look like this, which is in vivo in a rat running around. There are a whole set of electrodes of different depths. What you're seeing here is a slow rhythm, and nested inside of it is a much faster rhythm. So what we do with the information that we get like this is to use some version of Fourier analysis that gives you power in different frequencies, and from that one gets what's called a power spectral density map. That's what this is. If in the Fourier analysis, one is averaging over a longish period of time, where longish can mean, say, a minute, generally that power spectral density will be a 1/f curve. If one is averaging over something much shorter, like a second or 2 seconds, then you start seeing other frequencies. And here you see the low frequency has been cut off, but there's a bump around 40 hertz. I think it's more than that. It's a bump at a fairly high range. And all we mean by a rhythm is that it's a peak in the spectral power. So it's really good to look at the raw data and see that you're actually seeing something that looks like a rhythm. People have been looking at this for about almost 100 years now, and when they first started, they started giving names to different bands. So each one of these names actually refers to a frequency, where the signal is found in the brain, and the behavioral circumstances. So the famous first one, the alpha, in humans, it's about 9 to 11 hertz, back of the skull, with eyes closed. We now understand that this is far, far too simple, that any given frequency range can now be found in many different parts of the brain, with many different mechanisms, both mathematical and physiological, but nobody's come up with a better way of classifying these rhythms. So I'm going to use the usual names, but tell you more about it as I go along. So since these rhythms are kind of squishy, with people arguing about how to measure them, why should we care about them? And my answer is that there's a huge amount of data that connects rhythms with behavior of all sorts. For example, there's a repeatable and subtle change in dynamics that's correlated with tasks, very correlated finely in time. For example, when a rat is running down a track and needs to decide whether to go right or left, at that point, the brain rhythms change, and the correlations, the coherence between one part of the brain and another part of the brain also change exactly at that time. And this is not one example with nothing else, with no other kinds of data. We find that brain rhythms are associated with essentially everything we can call cognition, with sensing, with attention, with decisions, with memory, with motor planning. In every situation in which the brain is active, including sleep, you will see these brain rhythms associated with what's happening cognitively. And interestingly enough, and something that is one of my major interests at the moment, brain rhythms are associated with perhaps all forms of neurological diseases. That is, for any given neurological disease, there's a pathology in the rhythm. So one can ask the question of why should these rhythms be apparently so important in cognition and so important in brain disorders? And one answer to that is that these rhythms are highly associated with coordination of different parts of the brain. And by coordination, I don't mean just synchronization. I really mean a much more complex form of coordination that I'll be telling you about. So once one believes that, then it's more reasonable that if there are changes in rhythms, there are going to be changes in coordination, and those changes in coordination can prevent the normal kinds of interactions among parts of the brain that are necessary for normal cognition. So one of the major points that I'm going to be making is it's not just the frequencies of the rhythms that matter, but the underlying physiology that gives rise to those frequencies also matter a lot. So this is an example just to make very vivid how important coordination is in the brain. It comes from a paper about 10 years ago that was about working memory. And what you see on the right is left and right hemispheres of the brain, and every line has to do with changed coordination during this task. And the authors of the paper were looking at this kind of functional connectivity in different frequencies, in the alpha frequency, in the beta frequency, and the gamma frequency. And the major thing that I want to point out here is just how much brain real estate is being used during that one task. It's a huge part of the brain that's being used, and hence coordination is essential to the function here. I also want to point out that it's become fashionable to talk about the connectome, which are the physical connections between one place and another place. But the fact that these connections can depend on the signals that are going across those connections means that we really need to be thinking about not only the physical connections, but the dynamics associated with them. And for that, I coined the name the dynome. So I'm going to be telling you about the dynome. Okay, I have exactly one slide here that has any equations whatever on it, and this is it. So this is to tell you about the general mathematical framework that I'm going to be using that's going to help produce, that was used to produce all of the simulations that you're going to see. It plays the same role in neuroscience that the Navier-Stokes equation play in fluid mechanics. It's not just one equation, it's a whole collection of equations with a framework. And this framework is about looking at the voltage that goes across any given piece of a neuron at any given time. And since neurons are spread out in space, this equation, which is Kirchhoff's law, conservation of current, is a partial differential equation, but generally one breaks up the whole neuron into little bits called compartments. In simpler situations, you might have just one compartment. And then, in addition to the currents that go across the membrane, so this is an electric circuit theory, basically. You have the currents that go across, capacitance current, synapses, which provide more currents when you want to connect different neurons. And these synapses can be excitatory, which lead the cell to spike, or inhibitory, which steps on spikes, at least temporarily. So a critical thing to say is that unlike the first course you ever took in elementary physics, the currents themselves here, this is E equals IR, Ohm's law, solved for I. So the conductance, which is 1 over the resistance, is not a constant. It depends, the M and the H here are gates that open and close, and they depend on voltage, and they depend via their own differential equations. So when you put it all together, you have a highly nonlinear system, which has produced a lot of pleasurable work for a lot of mathematicians. Okay, I want to talk to you first about inhibition because it's going to come up in a lot of the examples I'm going to give. So this is an input to a cell which will push the voltage in a direction that keeps it from spiking. And here you see 10 cells, and they're not coupled to each other, but they've been initialized so that their spiking is as far apart from each other as possible. And at time 0, they all get an input. And notice that when the inhibition wears off, they all spike at roughly the same time. If you had another pulse, they would be almost exactly at the same time. So what's happening is this pulse of inhibition is driving all of these cells close to a critical point, an unstable critical point of the equations. And so when the inhibition is taken away, they all fire more or less together. And this is going to be important because I'm going to be talking about networks of excitatory and inhibitory cells, and they can synchronize if the inhibition is shared. So let's look at the simplest, I think, of the important brain rhythms. It's called pyramidal interneuron network gamma, or PING. And it was discovered in around 1997 in vitro. It's associated with cell assemblies, which I'll tell you a little more about. The easiest way to think about this mathematically is if the network has exactly one excitatory and one inhibitory cell. The excitatory cell fires, the extra excitation makes the inhibitory cell fire. It gives inhibition to the excitatory cell, which wears off after a period of time, and then it starts all over again. So this period depends on the decay time of inhibition. And you can see that there's nothing else in this network that has a longer time scale, which will be important to us. If you now have a whole lot of cells, here we have 400 E cells and 100 I cells, and you start them off completely randomly, even if the network is heterogeneous in its parameters and has very sparse coupling among the E's and I cells, it will self-organize into this gamma rhythm very, very quickly. The mathematics behind that is in a paper with Christoph Borgers. Okay, so now I want to tell you about why this can be important. And so the motivation of it is, the motivation of my telling you about it, at least here, comes from the idea of attention. Attention is a very complicated idea in neuroscience, but at least aspects of attention are thought to be mediated by competition. That is, if some input, bottom up or top down, is in some sense more important, not spelled out, than another one, then it will win. And so Desimone and Reynolds had a very important and influential paper about this. What their paper doesn't account for is the observation that essentially every time you have anything you can call attention, you see a whole lot of gamma. So what is this gamma actually doing? Well, one thing to add to what I just said is that attention seems to be associated with a more coherent input, by which I mean that the inputs can go on for a while, but they might be, the inputs from a whole lot of cells will come within a millisecond or two of one another. So this simulation that I have below here is going to be telling you about how more coherent inputs can lock out less coherent inputs, and thereby give you a winner for a competition. So the target is the simplest possible that will produce a gamma rhythm, one E and one I, and there are two inputs in it. In the first simulation here, the input is spiky, that means very coherent, and there's no input at all for the second one, it's 0. And the output of the E's and the I's are just what you would expect. They spike every time there's an input. The input goes to I as well, but it's smaller, and I'm just showing you the input to the E cell. So that's not surprising. What's surprising is the second one. We have an input that's gamma. We have another input that's a slightly different frequency. It has exactly the same current as this one. And we now look at what the output is. And here's what the surprise is. The output is essentially identical to the first one. So that the coherence of this first one is protecting against another input. It's simply shunted out. So how does this happen? And this gets to my point that what rhythms do in the nervous system isn't just dependent on frequency, it's dependent on the underlying physiology. And in this case, it's dependent on the fact that that this target has feedback inhibition. So to illustrate, the mathematics of this was done in a paper with John Cannon. So this is just illustrating what's happening here. We start with an input that's the same as the gamma input we had before, and now we have a huge other one, a huge distractor. Its average is even larger than this. Its amplitude is very high, and not wholly surprising, there is an effect on the output. You see, it's very different from the output we had before. So this one is making its mark. However, if we increase the feedback inhibition, then this one is totally ignored again. And we get back the output we had before. So this is why the physiology really matters. The frequency also matters. What is needed to make this happen is that the time scale of the decay of inhibition should be on the same time scale as the oscillation. Okay, I'm now going on to story number 2. I have 4 short stories for you, the middle 2 being quite short. I've just been talking about gamma now. But it's well known that in the nervous system, so gamma is about 30 to 80. It's well known that in the nervous system, many rhythms can be seen in exactly the same signal. And a pair of rhythms that are often seen together are gamma and theta. I actually showed you a picture of this back at the beginning. I showed you a fast rhythm nested in a slower rhythm, and that was gamma and theta. So our next question is, how do these different rhythms, which are going on at the same time, interact with one another? And my student, Yuja Zhou, did some work with others in our group too, did some work thinking about a relatively simple example of interactions in which the input was a gamma and theta, but the target also has a gamma and a theta in it. The target is only one cell, but it has an autopse, that means a synapse onto itself, which is inhibition, and it has a time scale, and that time scale is associated, as in ping, with gamma. But we also add something that gives a theta time scale. That's a funny current called an M current. It's an outward current, so it's kind of an intrinsic inhibition, but it turns on when the cell gets excitation. And its time constants are consistent with producing the theta. So you now have gamma and theta both in the target and in the input. And the question is, do these timescales interact? And if so, in what way? Okay. So, yes, they interact. And we found something that initially surprised us and then didn't surprise us. It turns out that when you have a theta input with a gamma input, it actually gets in the way of the system locking to the gamma. So if one needs phase locking, for example, in plasticity, it's really important to have that fine timing. If you have a gamma and a theta coming into a simple network that doesn't have that M current in it, it can foul up this locking. And that's what's being shown here. So we have a gamma input, we have a theta input here, and the black is the voltages in time, and you see that it's following the theta, but it's totally ignoring the gamma. Now we look at what happens if we add an M current into this. To make the comparison fair, we have to normalize it. The M current is an outward current, so you have to balance it with some tonic current, some constant non-oscillatory thing to make them have the same excitability. And when you do that, you find that with the gamma and the theta input, the output does follow the theta in bursts, but it also follows, in fine timing, it follows the gamma. So it follows the theta and it follows the gamma. So how does this happen? It's because the M current is homeostatic. What that means is that it's opposing, whenever there's excitation, it's opposing it by having this intrinsic inhibition. And you can see this opposition by looking here at what the M current is. So every time the voltage, every time the theta gets to its largest point, the M current, which is plotted downward because it's outward, is balancing it out. And that balance shows up in the voltage here. And that subtraction of this baseline here is what's allowing for the fine timing. I don't think this is obvious until you start thinking about what currents do. Okay, here is another story, short, it will be only one slide. And it's a little bit like the last story in that it's about control of phase locking, and it's also about the effects of this intrinsic inhibition or outward currents that themselves have time scales in it. So it's a little more complicated than the previous one because in addition to having this M current that I was talking about, which is an outward current, with a theta time scale of recovery and having synaptic inhibition, we have another kind of intrinsic inhibition from what's known as a calcium-dependent potassium current. You don't really have to know what that is. The point is it's outward and it's considerably slower than an M current. It's on a delta time scale, which is about 1 to 4 hertz. Okay, so what was the point of doing this project, which I'm about to tell you about? It has to do with speech, in particular about comprehension of speech, which requires segmentation of syllables. It turns out that speech is comprehensible if syllables arise between about 1 hertz and 9 hertz. Slower or faster, it's really hard for a person to understand them. And of course, the length of the syllable changes from syllable to syllable. So the average, although the average is about 3 hertz, the actual flow of syllables is changing throughout between about 1 and 9 hertz. So what role do oscillators play in this? It's believed that there's a natural frequency in the auditory cortex, which is about 7 hertz, which is significantly higher than most of these syllables. That somehow or other, that locking to, and there are theories about this I'm not going to go into, that the brain needs to be able to identify, segment out those syllables, and it uses the oscillator to do this. There's an immediate issue here because where the frequencies that we care about are lower than this intrinsic rate, which is about 7. And if the oscillation is to be governed by something coming in, you have to make sure that the target isn't just firing by itself without waiting. So Ben Pittman-Paletta of my group is almost finished with the paper now, which looked at whether you can flexibly get locking to these kind of speech inputs. And he worked with, there are actually 6, I'm only showing you 5 here, models. Some of them have just 1 cell in it, others have 1 cell plus an inhibitory cell attached to it. This one has an M current, this one has synaptic inhibition from this other cell. This one has the inhibition and the slow outward current. This one has inhibition and the M current. This one has just the 2 intrinsic inhibitions. And let me tell you what you're seeing here. This is input frequency. This is strength of the input frequency. Every one of these is a different simulation with different input frequencies or strengths. And what's being plotted here in color is what's known as a phase locking value. So when you don't have real complete phase locking, there are statistics that enable you to say how much locking there actually is. And that's what's being plotted here. So these 5 different models are being put in order of how well they do at being able to lock to these low frequencies. And the best one, the worst one is the M current alone. The best one is the M current with the much slower current. We have some mathematics that shows what's happening here, but it's complicated. I'm not giving it to you. There are some interesting multiple time scale analyses here, but they really do interact with one another. And what's going on is that the ability to lock to these slow frequency comes from a buildup of these outward currents, this inhibition, which prevents a spike, an intrinsic spike before the next output. So once again, you see that the physiology really matters. Interactions of time scales really matter. And here you get the most flexible response to not only to, it's a larger response in that over a longer range of frequencies you get good locking, but also it turns out that's the best at taking an input, and we've done it with inputs from speech, which has a variety of, in which the time from syllable to syllable keeps changing. This will lock onto it very well. Okay, last of my 4 examples. So far In each of these examples, I've been basically talking to you about very small networks, 1 cell, 2 cells. And here I want to go into the kind of complication that comes about when you have the kind of large networks that the brain is actually made up of. So here, the physiology is going to be simpler. We don't have the funny currents. We just have standard Hodgkin-Huxley. It is an EI network, and we're going to talk about what large networks can do that small networks can't, which is they can be heterogeneous. That is, if you have a whole lot of excitatory cells, they may have different parameters. They may have different baseline excitation. They may have different connectivities to the eye cells. There are a lot of parameters in Hodgkin-Huxley that you can change. And what we found was that heterogeneity, which some people might think might get in the way of phase locking, actually can help a network to phase lock very strongly. We're using extremely weak inputs here. Most of them are subthreshold. They could be a little above subthreshold. Okay, so first let's ignore the eye cells and just look at the E cells, which have periodic input from 0 Hertz up to about 70 Hertz. And they also have tonic inhibition that gives them a natural tonic excitation that gives them a natural frequency, and there's some noise here. So this is showing that if there's no noise at all, the output here, so this is the frequencies of the output, and the color is the power at those frequencies, that the output is basically the same as the natural frequency no matter what you put in. If, however, you add noise to this, allowing for something like stochastic resonance, now the network can actually synchronize from about 0 all the way up to 70. So this is surprising. I'll tell you a little about how it works in the next slide. So now let's put the eye cells back in again, and we try to get just noise. So this is without noise, this is with noise, E and I cell. And you notice that for no noise, it's basically what we've seen before. No matter what frequency you put in, you get out a natural frequency. But now if we put in noise, you get some locking to all of the other frequencies, but you still have a very strong locking of just the natural frequency. So noise is not enough to allow entrainment in these heterogeneous E to I networks. I'm sorry, it's not enough to allow locking in these homogeneous networks. So now I'm going to get to the heterogeneity and what it does. We looked at a lot of different kinds of heterogeneity, and the one that was the best was heterogeneity and connectivity from the E cells to the I cells. Meaning that some eye cells got a lot of input from E cells, some eye cells got very little input from E cells. And what you see is with increasing heterogeneity that you get less and less of the natural frequency and more of locking to everything from 0 up to 70. Those are harmonics you're seeing over there. Above and below the natural frequency. We tested a lot of different kinds of heterogeneity, and most of them do not significantly increase the range of entrainment. This is an increase of about a tenfold over entrainment just to the natural frequency. Tenfold, it's huge. Okay, so how does it happen? Well, for that, we have to go back to looking at phase locking value. So every one of the pixels here is a different simulation. Here is input frequency, and every one of these is every line, every horizontal bar is a different neuron. So what you see is that as the heterogeneity gets larger, different subsets of eye cells start responding to different inputs. And when you get to the highest amount of heterogeneity, basically almost from 0 up to 70, there's some subset of eye cells that will lock to the frequency that's coming in. The reason it can do it is they're heterogeneous and they have different levels of excitability that are coming from getting more or less excitation from the E cells. So the eye cells are able to respond to the frequency. And then the second point is that the eye cells alone, even in the absence of any input to the E cells, via that feedback inhibition, will make the E cells respond. So, so we're able, via the increased heterogeneity, to be able to get phase locking at very, very low input strengths across a huge range of frequencies. Okay, I'm now going to tell you what I said and then the takeaways. So, the focus of the talk was that brain rhythms are not quite oscillators. They produce oscillatory power, but they're not what we would call standard oscillators. They're not regular, they're squishy. And I gave you 4 examples of what these squishy objects can do. So the first one was about ping. It's just a simple EI network and how it creates a coherence filter that can lock out inputs that are not, using feedback inhibition, it locks out inputs that are not as coherent. The second story was about the fact that in the absence of the M current, a theta rhythm can prevent locking to a simultaneous gamma. But if you put in this homeostatic current, which is a kind of anti-resonance current, it subtracts out a current, a frequency, then you're able to get very fine locking to the gamma. The third one was about how intrinsic inhibition, outward currents, are able to allow cells to lock to slower frequencies and how they actually interact with one another, enabling locking to a large range of small frequencies, way below the natural frequency. And the last talk was about heterogeneity in this, a much larger and noisy network allows locking to a huge range of frequencies you would not get in a homogeneous network. So all of these things are going on simultaneously throughout the brain, and this is the tip of the iceberg. So what are my takeaway messages? First of all, as soon as you have a target that's more complicated than anything that's described by phase, like a limit cycle oscillator is, the dynamics of the forced oscillation has much more structure to it. It's not just all over the place, there's real structure but it goes beyond what we think of as the structure in a phase oscillator. And then there are a lot of other variables besides the phase that are really important. And these, like the feedback inhibition. These inputs affect variables besides the phase, especially the synaptic and the intrinsic inhibition. And so all these other variables can interact with one another, making it still more complicated. All of this happens even in one or two cells. But if you have large cells, they can be even large networks. They can be even more complex because of the heterogeneity. And they can produce different things because of the heterogeneity. So the bottom two takeaway messages I have is we have close to a complete theory of forced oscillators. Not quite, there's still open questions there. But the theory of forced networks is essentially at its beginning. So the bad news is that there's so much that we don't know yet about how input to a network where the input has complicated spectral properties and the target has complicated spectral properties, we're just beginning to understand how that works. And this is critical to the coordination throughout the entire brain. But the good news is, especially since this is a math conference, a dynamics conference, is that there's a lot of math left to do. Some of the math behind the examples I gave you has been done. A lot of it has not. Almost anywhere you look, there's a great math problem. So I want to leave you with a sense that understanding brain rhythms and their interactions is both important to neuroscience and can provide many open and interesting questions for mathematicians. And I thank you.
Thank you very much, Nancy. I'm sure there is a lot of virtual applause for this very nice talk. So, like I said in the chat to other attendees during your talk, I have forgotten to remind them that they could use the Q&A box, but some did. So there are already questions that have been typed. So I'm gonna direct these questions to you, and then we'll see if there are further questions. So the first question by Alan Champneys is, can you apply this theory to specific brain regions which have different functions and different wet lab data?
And the answer is absolutely, and we are. So we are working with many collaborators. I'm especially interested in abnormal brain dynamics. So one big project I'm working on has to do with anesthesia and how, in this case, we are basically looking at different parts and coordination among different parts of the neocortex and using data from both rats and non-human primates and the operating room, humans. So this is a very large project that's being coordinated by Emory Brown. And our main hypothesis is that what anesthetics do is hijack all the rhythms in the nervous system, making it impossible to coordinate adequately to have consciousness, that the loss of consciousness is a reflection of the hijacking of normal rhythms. And our data, we now have a lot of work, and the data supports that idea. Another big project that I'm working on is Parkinson's disease. And here we're talking about the basal ganglia in the subbrain, in the striatum in the basal ganglia. And we have, again, a lot of work on mice and monkeys and humans. And once again, it appears that the inability to move, which is the hallmark of Parkinson's disease, seems to be due to a hijacking of the normal beta rhythm, which is important for many different functions in the nervous system. And what happens in Parkinson's disease is that the beta takes over and doesn't allow other things to happen. So we have a whole bunch of projects that are associated with that. I'm working with Sabine Kastner on attention, and there the data is coming from LIP and FEF and V4, all parts of the neocortex, as monkeys are doing some task. And our gang is accounting for the rhythms that they're finding and the functional importance of those rhythms. And I could go on because there's a whole bunch more. But that gives you a sense. I love your question because I think it's really important that whatever theory we mathematicians do are as tightly tied as possible to data and working with experimentalists.
Okay, thanks for this very detailed answer. So we have a question from Simon Berman, who's asking, where does the noise appear in the heterogeneous EI networks? And is it simply white noise or some other kind?
We've used Poisson noise. We've tried other things, and in our hands, it doesn't make a difference. And it comes to the E cells, only the E cells.
Okay. I think Daniele had a question, so maybe he can unmute himself and ask it directly.
Yes, I do have a question that is related to Simon's questions, and it is does the heterogeneity have to be applied to a specific parameter to obtain what you see?
Absolutely not.
And does it
absolutely not? It's extremely robust. Well, I should say that the heterogeneity is very robust when one thinks about the heterogeneity between connectivity between the E cells and the I cells. You get some effect, but not as strong if you just have heterogeneity in the underlying excitation of the I cells. If you try doing heterogeneity in many of the others, the effect is very, very small.
Thank you.
And we think, as I said, that this is because with the heterogeneity in the E to I, we're able to, the frequency is able to pick out the right set of I cells that will play nicely with it. And those I cells can then entrain the E cells.
Thank you very much.
Okay, the next question for you, Nancy, is from Tushar Chauhan, who is asking, how do the neurons select which rhythm is selected and which one gets shunted?
Well, according to the mathematics we've done, well, first of all, that particular issue that I was talking about at the beginning, gamma filter, is only about gamma. The inputs are all around the gamma frequencies. So we have a very incomplete understanding of what happens when you have inputs that have multiple frequencies in it. So this was just locking out another gamma. It wasn't locking out a theta or a beta or a delta or an alpha. I gave you an example of what happens when you have inputs that are theta and gamma and how they can interact. This is an example of my statement that what we don't know is enormous. And it's a very, very general question of when you have a very, very complicated input, what does the output do with it? And this is a really simple target, which itself doesn't have much dynamics in it. But when you have a target that has heterogeneity and a whole lot of intrinsic currents, what it will do with the inputs it gets is anybody's guess now. But that to me is the central mathematical issue. I mean, you phrased it as basically, what does it lock out? But I don't think it always locks out everything. It takes something from here and something from there and does things with it, but what does it do? It's a whole field of mathematics that barely exists yet.
Okay, thank you.
Which I hope our listeners will feel interested in adding to.
I'm sure some of them will. So there are a couple more questions that we have the time to ask you. One
That's
from
wonderful.
Kale Timperi. I'm sorry if I mispronounce your name, Kale. So he or she says, great talk, thank you. Are there any particular insights that could be obtained for understanding the mechanisms of sleep using your approach?
Absolutely. And indeed, we are, because sleep has some connections with anesthesia, we are indeed working with sleep as well as with anesthesia at the moment. So I would say that a critical thing that ties sleep to what I'm talking about has to do with the interneurons. I've mainly been talking about the simplest kind of interneuron, known as a fast spiking interneuron, but there are actually at least 3 different major kinds of interneurons. And one of the things I'm really interested in, because it seems to be associated with a lot of problems with brain disorders, is the interaction of different kinds of interneurons, known as fast spiking interneurons, somatostatin interneurons, and so-called vasoactive intestinal peptide interneurons. And it turns out that, for one thing, circadian rhythms, which are associated with sleep, seem to be associated with this last kind of interneuron known as the vasoactive intestinal peptide ones. And many of the, so there's also a lot of work that's been done already on modeling sleep. What I think, and including rhythms, because the circadian rhythms are so important for sleep. But I think what our group is beginning to add to it is thinking about the specifics of the physiology of these multiple kinds of interneurons and how that plays a role in coordination of sleep. We have a paper that's almost finished on that, that involves VIP neurons and sleep.
Sounds great. I think in the interest of time, we're going to take one more question because then we have this social event, but I should mention that you said you might be able to attend some sessions and so interact.
If I can figure out how to do it, I will be there.
I'll be happy too. So one final question live for you, Nancy, by Ian Hunter. Thanks for the wonderful talk. Do you think it will be productive to study forced phase amplitude type networks rather than just weakly coupled limit cycle oscillators? Is it more productive to study forced Wilson-Cohen type models?
Okay, so I think it is not productive to study forced Wilson-Cohen models because it leaves out all of the physiology. And I think that the physiology is so central to what's going to happen. So if there are certain kinds of rhythms like ping that use very simple physiology And Wilson-Cowan may be able to get at some of it, although it doesn't have the timescales associated with inhibition. When you start getting to more complicated rhythms, even if you produce the rhythms, even if you produce something oscillatory at the rhythm you're interested in, you're producing it for a wholly different reason than the brain is. So if you study how it responds to inputs, you're not studying anything associated with the brain anymore. So I think it's really important, although we have simple, it's good to use simple models if one can, to make sure that that what's being looked at is not so simple that it's wholly irrelevant to the question that you're trying to answer. And if we're trying to understand how a target with a lot of different currents in it and heterogeneity and all of these things respond to inputs, then modeling the target by something much, much simpler than that is just going to be irrelevant. If it leaves out everything that matters, it's going to be irrelevant to that question, maybe not irrelevant to other questions.